Hi. Good evening, everyone. If you just joined us, can you please put your phone on mute? I would like to welcome you all, and we'll start the program now as we see about 22 people have joined, and we are also live on Facebook. I'm going to hand it over to Dr. Arlene Myers, and we'll kick off the program just in a couple seconds. So I'm going to put myself on mute, and Arlene, why don't you kick it away?
Thanks very much, Dhruv. And I really want to say thank you to all the participants on the panel and our collaborators at IBE. Our SoPE network is growing substantially for a number of reasons, as you can imagine. And if you are interested in getting an idea to a patient, or if you're interested in helping someone who is, that's really the mission of what we're trying to accomplish. We're an open innovation network, so you do not have to be a health professional to join us. Anyone can join. We firmly believe that sick care cannot be fixed from inside, so we really need collaboration from various industries, from various people, from various functional areas to make what I think we all agree is a sick system to make it better. So if you're interested in joining us, our website is at www.soapnet.org. And once again, thank you for tuning in for what I'm sure you'll find to be a very interesting presentation. So Kurt, do you want to take it from here?
Sure. Thank you, Arlen. I appreciate the introduction. Here, let me just share my screen here. And can everyone see my screen?
No, not yet, Kurt.
Okay, here, 1 second. Here, we practice this. All righty.
Looking good.
Now there we go. So I thank you for this opportunity to speak to the SoPE membership this evening. I am Kurt Sprouse, the president and one of the founders of the IBE. And what I'll do is I'll take you through a brief overview of why the IBE was created and what we're doing. And then what I'll do is I'll turn it over to 3 of my colleagues. Ratmer will lead off with presenting a company that he founded. Ratmer is one of our IB alumni. Tony Manning, our chairman, will share with you how he utilizes the IB as a resource with his organization, Momenta Pharmaceuticals. And then we'll conclude with a presentation by Tammy Dorsey, again, another IB alumni who has founded a company, and she'll share with you her story and journey. So the IB is really focused on this idea of translation. And there's a tremendous challenge that exists in translation driven by 2 fundamental problems. The knowledge gap, which is that many innovators, people that come from technical or clinical backgrounds, just don't have the insight into how to take an idea from concept to market. They're very adept at understanding the technical nature of their chemistry, biology, engineering, the clinical aspect of delivering care, but taking that concept and making it a business idea that's viable is challenging. So the knowledge gap represents one significant issue. The other is obviously the funding gap, aligning capital with early staged ideas that have many times not yet been proven. It's a complicated environment, and navigating that environment, investment, regulatory, the development, manufacturing, testing, approval process is quite complicated. And so what the IB was created to do is help with that particular ecosystem and how to navigate it. Some of the factors that have led to this challenge are that there is a decline in R&D productivity. I think that that's driven primarily from the standpoint that all the easy problems have been solved. The challenges before us, and I think COVID is a great example of that, represent significant challenges on many different levels, and the timelines that are that we need to abide by to be successful, not only from a health perspective, but also from an economic perspective, also represent challenges that are significant and more challenging than they had been in the past because of those economic factors and the difficulty of the science or technical challenge before us. So again, we look at the process, and it's a long, drawn-out process. The agency is reacting to new therapies, new concepts, and in many times, the way that we will test and prove the safety and efficacy of these ideas are unknown because they are such new, innovative ideas. So again, it's not just going through a process that's well-defined, but it's creating a process around new, innovative ideas that are being presented to the agency that represents another barrier for success. We also find that the cost of failures is significant. We're seeing an increased cost of failures at all levels, both in preclinical through phase 1, 2, and 3. And the cost per failure is exponentially higher and has grown exponentially over the last few years, as you can see from this chart. So the gaps exist because we are very focused on the R, but not so much on the D. Industry as a whole, the ability to transition from the research to the development is challenging. The investment community has looked to invest in more predictable outcomes and moved away from the translational space. There's still quite a bit of money there, but the interest in VC and in investing in those early stage ideas is much more challenging than it had been. The biopharma players are not set up to do that early stage work. So that is another challenge that is something that's added to this problem. And most innovative ideas are just that. They're ideas, but they're not yet ready to become companies. So again, how do we deal with that? We know what makes up a good idea. We know what the factors are that contribute to a successful idea, but the pathway to achieving that is sometimes difficult, as I've mentioned. Having something that's unique, that's going to solve an unmet need, that's reproducible science, novel, defining a clear path by which that idea is moved forward, and creating value that allows us to raise capital to achieve each of those inflection points that will allow us to progress in this process is something that many innovators have not gone through. And it isn't like you can define it and it's always a standard process by which you achieve success. That dynamic changes as the system changes and as the company encounters different hurdles. So back in 2015 and 16, we assembled our first group of individuals in Boston and Cambridge to talk about how we would solve this problem. And right here you see an article that was published by, that featured Tony Manning talking about this idea of creating an asset factory, an organization that would take innovation and allow the innovation to prosper and advance in a more reasonable and predictable way. And that was the really start of this idea of the IBE. Tony Manning and I joined with Henry Skinner, who's with Techla Ventures, Greg Fairbrothers, who ran the entrepreneurial program at Dartmouth for 14 years, and Chuck Yon, who is general counsel for Checkmate Pharmaceuticals, to form this entity that we call the Institute for Biomedical Entrepreneurship. And if you look over the time that we've established this particular group, which focuses on a couple factors, education, support, funding, we've attracted a number of industry players. You can see here, pharma, biotech companies, I've named just a few, device companies. But what we've also been able to do is create a number of successful initiatives, both within industry and out of the academic institutions themselves. And I've identified a few of them for you. And we're going to be fortunate enough to hear from 3 of those individuals this evening. How we solve that problem is a mix of education, networking, capital, and support. The educational program is basically a 5-day intensive graduate level program whereby we introduce the individual to the concepts that allow us to introduce them to the spectrum of knowledge necessary to advance an idea, surround them with a network of faculty members who are experienced, who become their support system and network, and then provide them access to capital and funding mechanisms to help them to gain the resources they need to establish their ideas and start to prove their concepts. So hopefully this brief introduction gives you a better idea of what we're doing. I've provided our website. There's a lot more information and case studies available on the website. And with that, what I'm going to do now is turn it over to Ratmir and have him talk a little bit about his company, 48 Hour Discovery, and the work that they're doing and how that was supported by the IB initiative. So Ratmir, if you want to jump in.
I'm going to jump in and share my screen. Can you see me?
Yes, we can.
Okay. All right. I'm going to go through a couple of slides I put together describing the company 48 Hour Discovery, founded shortly after my attendance of the IB, one of the first IB sessions in Boston. So the title is the same as in the program. So this is a case study of molecular screening using genetically encoded phage display molecules. And if you have any further questions that are not answered in this after this presentation, I encourage you to contact me at ratmir@48hourdiscovery.com. So I wanted to start just by introducing very globally the concept of what molecular discovery is and why is it important. And maybe I want to make a very broad statement that molecular discovery is basically an endless market opportunity. That every year there's going to be new molecular targets coming out of the fundamental studies in medicine. These molecular targets would be important in development of new drug leads. And oftentimes when molecular target is discovered by medicinal genomic proteomic studies, first thing we're going to need in order to develop an IND-enabling drug lead is to have a concept of molecular ligand that can interact with the molecular target. Majority of companies that start in the development of pharmaceuticals start from the concept of ligand for which they will develop a series of molecules, target for which they will develop a series of molecular ligands, which then through a series of trials will be converted to a drug. So I'm going to use the word ligand, and those of you who are familiar with it, I'm sorry for excessive introductions, and those of you who are not, just think of the ligand that as a key entity from which many downstream values originate. So one of them is IND-enabling drug lead, and then any other molecular entities, such as diagnostics, binding agents, handles for diagnostic devices, for imaging, for delivery, and even for culture. All of these originate from a very fundamental interaction between ligand and a receptor. So let's say you're a brand new company working in a space of cardiovascular diseases or COVID or anything else, and you say, we've came across this very important molecular target for which we need rapidly to get a series of molecules that will bind, actuate, block this target, and will allow us to proceed forward into development of our product. So what you want to do is, ideally you can just Google it. And if you cannot Google it, you want to develop it at the speed of Google. That's how I like to say it. So you want to send off this target somewhere, have someone process this target against many, many different molecules and come back with the list of these molecules and say, here's the list of molecules that bind to a target. Take this list, take it into your development, pick the ones that work for you, and go ahead, download this list from the cloud, from the web, from our depository, and you use it for whichever added value applications you want. Therapeutic diagnostics, detection monitoring, capture purification, or if it's a new market in which you want to add value by having access to target ligand interactions, again, we want to be able to serve that. So in a way, we see this opportunity of creating new molecular information for custom molecular targets and spur all sorts of business opportunities out of it. And the concept of 48-hour discovery is very simple. Give us the target, we will give you a list of ligands that bind to this target very quickly, ideally in 48 hours, which is the future long-term goal. So 48-hour discovery came about 3 years ago. We're 3 and a change years old. We have 10 full-time employees. We're located in Edmonton, Alberta, in Canada. We're fully integrated company with wet lab and IT infrastructure. So I like to compare it to early Apple, where we develop both the hardware, we develop our own molecular universes, and the software, cloud-based analysis of these universes in order to give us rapid decision-making. And we've been successful in raising all sorts of funds, direct from clients in form of contracts. We work with top 4 out of top 20 pharmaceutical companies that unfortunately I cannot name here. We have 16 other contracts from other entities, both academics and industrial. We supplement these contracts with lots of Canadian grant funding, which is extremely nurturing soil for entrepreneurship. And we have been successful in closing a round of investment recently, last year. And our mode of operation, as I said, is a combination of unique molecular universes in which we perform discoveries. And these are billion with a B, billion different molecules that we can rapidly search through, and then use custom analysis software that can give us decision making and say, these are the lists of molecules that you want. Few more technical details for those of you who know molecular landscape. So if you think of new molecular discoveries, they can range from ligands that are big and destined for therapeutic, biological-like therapeutics. So these are antibodies, large molecules from which you can develop antibody-based drugs, or antibody-based capture agents, or antibody-based lateral flow detection strips, et cetera, et cetera. So antibody-based market is very huge and great. And there is an opposite market of small molecules. These are oftentimes small molecules that are being developed into pharmaceutical drugs. We actually operate right in between these two landscapes, and I can explain you why. For aficionados in the business, the biologicals and chemical, these are sort of two separate worlds. Chemical drugs need to be made chemically, but biological drugs can be developed through biological processes. We actually pick the sweet spot where we can use biological techniques like selection, evolution, direct evolution to evolve these entities, but we make molecules that are small enough to still be synthesized through a chemical synthetic process. So, so sort of bridging the advantages of both. And slightly different view of the same landscape between biological drugs that has about 300 billion annual sales and a small molecule that has over $1 trillion in annual sales. There exists this intermediate area that I'm going to refer to as peptide-derived therapeutics. Peptide-derived therapeutics is a rapidly emerging new field that, again, combines the benefit of both worlds. The beauty of small molecules that these are the core of pretty much every orally available drug on the market. The downside of small molecules is they can go only after so-called druggable targets, which I'm happy to explain what they are. The beauty of biological drugs, such as antibodies, is that they can target any druggable or non-druggable targets, however, in only very particular extracellular space, and these are mandatory injectables. And through the analysis of existing approved drugs and drugs in clinical trials, you can see that peptide-derived therapeutics actually, they span both advantages. They can go after extracellular and intracellular targets. They can be formulated as injectables, but there's also evidence for oral formulation and so on and so forth. So this domain that bridges two length scales of therapeutics, it also bridges two very important advantages in the modality of delivery and the type of molecules that you can go after. It's a growing market. It's still a fairly small segment compared to the others, but this is the focus of our company, and we're striving to become the world's supplier of translation-ready, orally stable leads for market of peptide-derived therapeutics. And one of the internal foci is foci on GI disorders, so Crohn's colitis and microbiome modulation, which is actually perfectly suited for the PDT market. How does it work? Is this a fantasy? Is this a real world, or is this just fantasy? Right? How do we make these genetically encoded spaces? And is this just something we came up on a fly, or has this been around forever? And those of you who know the area a little bit, you recognize the word phage or phage display, very well-known technology that received Nobel Prize 2 years ago. It's called phage display. Its ability to display a molecule on the surface of bacteriophage. These bacteriophages can be mixed in one flask. You can have millions or billions of these molecules sitting in one flask containing a barcode. This technology is not only Nobel Prize winning, but it's 35 years or almost 30 years old. It's off the patent. And really, it's just a commodity that you cannot build a company around. But we see it as a valuable starting material that we can take and convert instantly into yet another space that has the same property. It sits in the same flask. We have billions of molecules sitting in the same flask with the encoding barcode. But now these molecules have properties that have not been seen before. So this is a brand new sort of matrix to matrix transformation. Billion of starting material to billion products, and all these products have interesting drug-like properties that I'll show you in the next 1 or 2 slides. For example, peptides are very uninteresting, they're unstable, and they're readily digestible in your stomach. Molecules that we synthesize are stable both conformationally, and they're stable to digestion. You can eat them, they'll pass through your GI. Many of them will pass through your GI without a change, without degradation. It's hard to discover something meaningful from a space of peptides due to their flexibility. It's much easier to discover from these constrained bicyclic peptides due to their rigidity, and so on and so forth. It's a one-step value addition to billion compounds at once. And we deal in these chemical spaces billion at a time. We made a billion of these, billion of these, billion of others, billion of others. And our company essentially as a fridge was the couple of tubes where we store these billion scale libraries and from which we do discovery. You can compare these to sort of sub-internet libraries in which we do the search. And I'm happy to explain how we make decisions when to search in this space versus that space versus the others. A head-to-head comparison with somebody who's already been in this field. A couple of companies engage on this idea of genetically encoded discovery as well as genetically encoded discovery from peptide-derived therapeutics. An example of that company is Bicyclic Therapeutics, started in Cambridge, not Massachusetts, the other Cambridge, and they underwent IPO in 2019 after about 9 years on the market. It was approximately $50 million IPO, and their core structure with which to operate is very similar to ours, constrained bicyclic architecture. We employ a different chemistry. We change the chemistry style. We change the architectural structure of these molecules. We create a new chemical and IP space. We improve the versatility and structural stability. And in head-to-head comparison, we see that the molecules that are developed in one of our sub-libraries are at least 10 times more stable. Some of these molecules are 10 times more stable to proteolytic conditions when compared to the bicyclic therapeutic structures. Next sort of litmus paper is, do these discoveries work for targets that are considered difficult in the business of drug discovery? We took a protein-protein interaction involved in tumor progression and in the maintenance of tumor stem cells. And we picked a particular member of the family called nodal, which is performing protein-protein interaction on a cell surface and stimulates a particular downstream responses. So what you're looking at is sort of anonymized structure of the compounds we discovered to bind to the protein nodal, and showing how these compounds can actually suppress the downstream signaling induced by nodal directly in a cell culture in a culture medium after multiple days. So these compounds were discovered from aforementioned bicyclic chemically modified libraries that have ability to antagonize important cancer stem cell signaling, and they're stable in digestive medium for multiple days. So they check multiple checkpoints that tells us that this is a promising lead for next step in drug development. And most important, we operate on a principle, if success happens, we want to be able to repeat the success over and over and over again. So one of the visions of 48-hour discovery is, again, something that I drew at the very beginning. The pipeline of target comes in, information comes out, and information comes out beaming into the cloud space and available to the client to evaluate, to list, to arrange the cluster and make a decision that these are the ligands we want to make. Those of you who want to know a bit more about technicalities of the process, I can say that we do multi-round screening, we do affinity maturation. Every standard technology that you would hear in the biologics discovery and development, we engage here, but we apply it to discovery of these sort of chemical mid-size drugs. So what do the clients see when they work with us? We try to look on the outside kind of like a typical FedEx. You drop off your package and you sit back and relax with your phone, and periodically you refresh it and you see that your projects have completed through panning procedure, through the sequencing procedure, through data analysis procedure. Now it's done. Now you can go check your data on the cloud. Here it is. Here are all the files that we've seen in the sequencing, and here's the particular view of particular files that you can open, examine, and make a decision. These are actually real screenshots from 48-hour discovery cloud. The four timelines that I'm overlapping, these are 4 different time lapses from 4 different projects. So you see that some of our projects are ongoing, some of them are close to completion, and some of them are sort of just starting. Curtis asked me specifically to not go too much into details, but focus more on the role of IBE. So here comes the slide that's probably the most important in this talk. 48-hour discovery more or less exists because I happen to be at the IBE at the right time. And I've been working in this area for 10 years as an academic. I mean, we dabble around all sorts of papers and try all sorts of cute things in the lab, but November 2016, Curtis invited me to come to IBE and listen to this very intense 5-day workshop where I was more or less forced to come up with a business plan around some idea. Very interesting, the idea, it grew into something, and when I came back, my eyes were glowing, my hands were shaking, I said, we need to do something. And a few months later, like literally just after the Christmas, we went on and we incorporated the company, and the rest is history. A few months after incorporation, the 3 other co-founders, I sent them back to the IBE and said, you gotta go there, you have to listen through this. So this is the team of scientists that kind of came in on board as co-founders, and all of them got up to speed with business lingo, with the mission, vision, value, and everything else to come back and continue growing the company. And many of the advisors that were in the room at the time of IBE, so including Curtis and Richard Gabriel, they became our sort of monthly advisors. Once in a while, we'd have phone calls discussing the progress. Curtis recently generously accepted to be one of the board members of the company. And with Richard Gabriel, we did a lot of joint projects and grants. So in a way, the 3-year development leading to this point where we are now, we're extremely well supported by the the present IBE staff. Feeling I'm a little out of time, so I'm gonna skip to this slide. I think there's a question session after this. And again, this is my email. I'm gonna hold here for a few seconds that you can send the questions to if necessary. And I'll stop sharing the screen.
Okay, thank you, Radmer. So 48-hour discovery is a great example of an entrepreneurial startup that, with the assistance of the IBE, Radmer, and founders, have really been able to move the company forward. So I want to just spend a little bit of time to show you an example of how the IBE has actually impacted an existing company. And I'm going to describe a little bit about the interaction between Momenta Pharmaceuticals and the IBE. I assume you can see my screen here. A little bit about myself and Momenta Pharmaceuticals. So I'm the Chief Scientific Officer for Momenta Pharmaceuticals. I'm a biochemist by training, and following a time as a faculty member at Baylor College of Medicine, I entered the pharmaceutical biotech industry about 20 years ago now. So about 8 years ago, I took over research for Momenta to help transform the company, and I'll talk about how the IBE has been central to helping that transformation. So Momenta is a typical Cambridge biotech success story. So it was spun out of MIT with some venture money in the early 2000s, IPO'd, and actually has 2 FDA-approved products in the market. They're both complex generics. We actually pioneered a new pathway for drug approvals in the generic space. The company is well funded. We're well supported by the investment community with strong cash reserves, about 140 employees. We've transformed the company over the last 8 years to move from complex generics into developing therapies to treat rare immune-mediated disorders. That's my own particular background. We have 3 novel drug candidates in mid-stage clinical development, all rare orphan drugs with meeting very high unmet medical needs in large markets. We have a discovery technology platform that fuels our pipeline, and we're actually in the process now of beginning to forward integrate as a full US commercial organization. So this is a very established company. Why would a company like this need to use the IBE to train entrepreneurs? This is an entrepreneurial success. Well, I think this goes back to the evolution of the company. Early in the history of the company, when it was founded, the original entrepreneurs founded it on technology to create generic products for complex mixture drugs. I won't go into the technical details, but it required a certain set of analytical chemistry, physicochemical, and manufacturing skills that really were central to the approval of 2 products that our partner Sandoz commercializes and we still receive revenue on. The company evolved from there to take on a next challenge in developing biosimilars as the emerging legislation provided a path for the development and approval for biosimilars in the United States. And we've worked with both Baxter and Mylan on large multi-program collaborations, but this required the company to develop new skills in handling biological products, manufacturing products, biotech products using living cells, and other technologies. But about 8 years ago, I joined the company to transform it to its next stage, and that was really to the promise of developing truly novel drugs, therapeutics for high unmet medical needs. And we've successfully developed 3 clinical stage products now that the details of which are not important for this. So the evolution of the business has required an evolution in the skill sets within the company. And that's where the IBE comes in. So early in the company, the skill sets you needed for complex generics were really about thorough physicochemical characterization of molecules. You didn't need to be an entrepreneur because you were copying an already approved therapy. The innovation was around the technology to be able to characterize and copy and manufacture reproducibly that generic copy. As we moved into biosimilars, again, not classical bio-entrepreneurship per se, because what you need to understand there are the technologies for product manufacturing, modification, engineering, producing biologic drugs, at 20,000 liter scale. It's when we moved into the novel drug area where there really was a growing need for these entrepreneurial skills of scientists to identify novel therapeutic targets for high unmet medical diseases, to develop novel approaches to modulate those targets, to move those products forward through the development phase. And Momenta took the approach, rather than going out and hiring experts, we decided to build from within for a couple of reasons. The first is you can't just go out and hire bio entrepreneurs who have exquisite skill sets in identifying novel product opportunities. As Kurt said, there's this gap that exists. We as scientists were never trained in bio-entrepreneurship. We were trained in deep technical and scientific knowledge. And so many of the skills and understandings you need to be able to develop a novel product idea and move it forward through value creation steps, those people just don't exist out there. So I decided to train the people that we had, who all had an extreme technical skills, who were all highly motivated, who all understood life in biotech. And what we did was that we actually partnered with the IBE. And so over the course of about 3 to 4 years, I sent 11 or 12 scientists from our discovery group now, individuals who were selected because of their high potential, because of their skill sets and their potential to become what I would call entrepreneurs in residence. People who would champion a novel product idea internally with funding from the company, just like they would if they were trying to build their own company on the outside. We sent about 2 scientists per course to the IBE training over a 3 to 4 year period. They took their own internal product ideas with them. We anonymized them appropriately to maintain confidentiality and maintain any proprietary knowledge. At the IBE, the actual target itself and the biology is not as important as the idea and the plan on how you're going to develop it. And so we were able to send our scientists with really meaty ideas to the course where they worked on their idea and learned all the components of putting together what I call a product pitch. Those scientists came back and essentially made their investment pitch to our executive management team. And from that work, we've actually had 7 novel product discovery programs funded. At the beginning of this year, we actually announced the selection of a new product candidate that we're taking to the clinic, which was actually one of those novel product ideas. And what I've seen internally is a real transformation of my basic research scientists into drug hunters. And so I think the IBE has been a great venue for training, for mentoring, for support, not just for academic or clinical colleagues who have good ideas and want to begin a company, but actually for scientists who even work within the industry to hone their skills and provide the education they never had to be able to help to get novel products in the hands of the patients who really need them. So the IBE has been able to help out both with forming new companies as well as enriching the pipelines of companies that exist. With that, I will stop, and I'll now hand it over to Tammy Dorsey to give you another example of that not a company-driven idea, but of a young entrepreneur with an idea who is starting her own company. So Tammy, over to you.
Great. Thank you very much. And thank you all for, let me get my screen shared here real quick. Let's see here. There we go. Can you guys see that?
Yep, we can see.
Great. Well, thank you for your time and joining us today and letting me share my story with how the IBE has been the bridge for me to where I am at today. And a little background on myself. I'm Tammy Dorsey. I have a biomedical engineering degree and a master's of innovation design, and I hail from Kansas, and I had a great foundation to come up with ideas and really innovate from a disruptive point of view. But the only drawback is I had no instruction on what to do with these ideas. So starting with Prenatal Hope, I had this idea that I wanted to help recover the US as a safer place of birth. The only thing is I was spinning my wheels, and I did that for about a year before I was able to be introduced to the IBE. And what that is like, you know, the audience that we have here, you're all the Society of Entrepreneurs, Physician Entrepreneurs. It can be a little bumpy, and it can be much smoother than this. It doesn't have to be as messy as this. The IBE for me, as a recent alumni, has immediate takeaways that you can apply with actualizing steps for the commercialization of this. It was a huge leg up in understanding the regulatory paths that are behind many medical devices, med tech, biotech. Whether your idea be a pharmaceutical or a medical device such as mine, the IBE is great for applying all of these entrepreneurial aspects that, quite frankly, are hard to find in many places. And the IBE works like oil in a machine. It keeps things moving. The daily engagements that you can have with the networking, the collaboration, all creates great traction. And one of the biggest things it will help you do is also help recognize where there might be pivots that are needed if things just aren't quite moving smoothly, or getting to the bottom of what those no's are or why you're being told no, with trying to close funding gaps and things like that. So for me, the process has been, you know, with the active engagement, absolutely critical, like a catalyst for my startup. And I'd like to share my startup with you as well, since we're all in different phases here. And my story is Prenatal Hope. You know, as a global superpower and state-of-the-art, or as a global superpower, we have state-of-the-art healthcare facilities and treatments. But one of the biggest issues we still face in the US is that we are the costliest, most dangerous place in the developed world to give birth. And one of the contributing factors to that is a doctor's inability to determine when a baby is in distress due to fetal acidosis. Now, essentially, this is a baby suffocating in the womb during active labor, and this can happen for a number of reasons, and it affects about 1 in 10 babies. This is why Prenatal Hope exists today, so that this doesn't have to happen to you or anyone else you know. I'm Tammy Dorsey, the founder and CEO of Prenatal Hope. I'm also the mom of 5, and this is my son. You see, it took 4 NICU doctors to save him. And the reason why is because they didn't have the tools they needed to know that he was in distress. They're using old technologies and external heart rate monitors, and his heart rate was normal the entire labor. You know, he's not alone. He's fortunate to have come out of it, but so many others are not so lucky. And with 4 million babies being born every year just in the United States, this is truly a need that needs to be met. So let's understand why this is happening exactly. The current protocol we have today, we have someone in active labor. The standard is the heart rate and contraction monitoring that's done. And from that, you either have a decision to either go with a natural birth or a C-section. Well, once the baby is delivered, then we can get this objective data that we needed. Well, but it turns out, we find out that it's wrong. Most of the time, the monitoring that was done during active labor doesn't match baby after birth. So we have this huge rate of error, and C-section rate, therefore, is going up. This is happening because doctors can't risk the liability. We would rather be safe than sorry, right? I mean, these are our children we're talking about. You know, and we look at this, you know, as Dr. Ender put it from Brigham Women's Health, heart rate does not correlate to distress. It does not directly correlate. It is subjective, it is interpretive, and it is quite often wrong. Doctors will order C-sections, and 9 times out of 10, the baby was fine. You know, we see, because of this, C-section rates across the world increasing in the US and beyond. The rate of increase is truly dramatic, and most of them are unnecessary as far as medically needed due to that non-reassuring fetal heart rate that's being detected with these external monitors. So looking at this objective data, wouldn't it be so much better if we could get that before birth so that we could avoid the train wreck, so that we could avoid the unnecessary C-section? And getting that data, that objective blood chemical analysis that is 98% accurate for fetal distress, would then lower the C-section rate. You know, we looked into what's out there, what has been done, why hasn't this been done? And clinical study after clinical study all weigh in the same, that pH is the gold standard for determining when a baby is in distress. So why hasn't this been done? Well, sensors have been made of glass, easy to break, too expensive, too much drift, not accurate enough. We have biofouling. There's time needed for blood analysis. Well, 15 years later in development, we now have an opportunity before us with pH ISPET sensors. And this corrects all the problems. You know, for the first time, we have a commercialization opportunity here to make a real disruptive change. The size of these pH sensors are such that they are now, you know, economical and cheap. We have contracted manufacturers that are currently making these pH sensors for other applications. And looking at the size of this, and first them being FDA approved, we have the cost effectiveness now with the microfabrication techniques that are being used. The accuracy, we have 0.01 accuracy on our sensors, and they're fast. So putting a team together to help me solve this huge unmet need was one of the things that the IBE was really essential. They not only look into what's needed for the entrepreneur to commercialize, you know, actualizing your steps, but they also do behavioral analysis. They'll help you understand where your weaknesses and strengths are, and what kind of people do you need to put around you to complement to make sure that you have the team that is the best at overcoming any obstacle. And so with that guidance, we've been able to build a team that has great multidisciplinary strengths. We've also been able to secure great representation, and also the regulatory analysis or regulatory assistance for our needs with the FDA. We were able to work with Hogan Lovells as well, who alone were responsible for 1/3 of all medical devices being approved last year alone. So with this team in hand and much daily involvement and iterations, we sought out over 200 plus OBGYNs and nurses that all had these great unmet needs. And we developed a platform technology with many different applications to solve these unmet needs. So this will give doctors and the nurses the tools they need to know when a baby's in distress in real time. And this will reduce those unnecessary C-sections. This will improve mother's health. This will reduce the cost and liability, but most importantly, this will help save lives. And we're not the only ones that feel like we've got some great ideas. We were most humbly excited to announce that we were featured in Entrepreneur magazine. Last year, we won one of the Most Fundable Company Awards. And this last January, we're featured at CES for Tech for a Better World. And we're also right now seeking opportunities with a Matter and Parkview Health partnership competition that we hope to do well in. You know, we've also had buy-in from our target end users as well. Our innovations are being seen as what's needed to break through the plateau that's keeping the US from getting our C-section rates to the World Health Organization's recommended rate. So our flagship product is the Vivo 2, and this will help doctors know when a baby is in distress in real time. But we're not just interested in helping to save the blue babies. We really wanted to take a look at these, the number of unreassuring heart rate tracings, as this is the standard and protocol. We're not trying to just, we don't want to replace everything that's there. We know that's a mountain that would be almost impossible to climb. We want to be an augmentation to what already exists. And so looking at those category tracings and the protocol that's behind those, they will look at the heart rate and use a fetal scalp electrode or fetal spiral electrode. Now this is what this looks like. It's a glass, I'm sorry, not glass, it's a catheter application that has a spiral on the end that is screwed into the baby's head. And this is just for direct heart rate. That's all that they're capturing with this device. Yet we have over 700,000 of these in use every year in the US alone. So Prenatal Hope looked at that and said, rather than just capturing the subjective heart rate, we can get the objective data when it's needed. And with that improvement on those spiral electrodes, get continuous pH monitoring as well. Now the patents on these have long expired, so we have a great licensing opportunity here as well. Now we took our platform technologies, our suite of products, and we had Hogan Lovells do a regulatory analysis for us. We need to know, with the confidence level of expertise that they have, what's the best way to approach FDA approval, what's the best way for market entry to reach our target market. And with that regulatory analysis, we've learned that the FDA would rather have incremental changes rather than just jumping straight to a Hail Mary that would probably trigger a huge de novo process and several million more dollars. So with that expert advice and counseling, we're looking at a stair-step waterfall approach with our devices to bring our solutions to help make that direct impact in the prenatal care industry. Our regulatory product timeline has also been helped map out. This was something that I was spinning my wheels on it was not going anywhere. We'll just say that. And it was the IBE that finally got us over the hump and making traction and making sense of it all. This is something that, you know, a lot of people will have to hire their own people just for the regulatory part alone. We're also very excited about our IP that we have as well, and have already submitted a response for our first office action for fast track approval. And looking at the marketing and the distribution opportunities we have NDAs with HealthJib Global and Cellmark Medical in Canada that are eagerly waiting for our products to be approved so that they can start the process to get these out. And we also have Blue Cross Blue Shield, an NDA with them, and they've been watching our progress, as they are very aware that just a 20% reduction in the current C-section rate alone would save the insurance companies over $6 billion annually. And with that, I would invite you to please join us in our mission. We're trying to help improve women's health care, reduce those unnecessary exorbitant procedures that are not needed, and help deliver healthier babies. And coming back to the IBE, you know, the process works. You have to put the time and effort into it, but it's going to be the catalyst that will, quite frankly, I don't know if we would be where we're at today. We would probably still be back on milestone number 1 rather than developed MVPs and ready for the FDA process. It closes the funding gap. It helps you de-risk. It helps teach you how to speak to investors. And, you know, they can even help with the hard part of knowing when to walk away when the deal's just not right. And those are those ugly truths that you need to hear for success. And my contact information, should you have any questions or want to reach out. I love paying things forward and giving back. And with that, I'll go ahead and give the screen back to Kurt.
Well, thanks, Tammy. That was a great presentation. Tony Ratner, very much appreciate you taking some time to go through your stories and how the IB was a factor in helping you advance your thinking and your ideas in your companies. You know, I just like to say to the audience tonight that we would welcome any questions you may have. We're happy, we have a little bit of time left this evening, and I know Dhruv was going to field questions from the audience that participated this evening. But you know, we're here to not only provide this educational platform, but a network. And I think what you've seen tonight, Tammy and Ratmir, they've become part of this family, which now numbers over 228 graduates of the program. And I showed you earlier in one of my slides, I didn't mention it, that we have identified over 80 startups that have come out of those 228 students. Many of them have never had any entrepreneurial training or background prior to attending the program. And the first program was launched, as Ratmir said, in November of 2016. He was actually in the very first class. So, you know, we're very proud of how far we've been able to come in such a short period of time, and we'd welcome, you know, interest, participation from those of you that are joined us this evening on the call. I don't know, Dhruv, are there any questions out there? And Tony, maybe you have some thoughts you'd like to share here as we conclude the discussion this evening?
Yeah, so this is Dhruv. So at this point, I think we would like to open up the floor, and anyone can put themselves on unmute. If you're having technical difficulties, just ask us in the chat, and we'll open up for Q&A. And but before we open it up for Q&A, Tony, any closing thoughts, or any of the other panelists?
No. Let's give the chance for some questions if there are any.
Sure. So Ivan, if you don't mind unmuting everyone, I'm not sure if attendees are able to do it on their own.
Sure, I can unmute everyone.
All right. So while we wait for the questions, I think, Tammy, I just want to say thank you for giving us the highlight. I heard you speak, I think back in October of last year, if I remember correctly.
You would like it because
it's amazing to see the journey from now,
I think that
going back to the October, and how much success you have achieved.
You know, so I mean, I don't mind it, but
on mute. If you okay, now we don't hear. So no, I think it's an amazing experience to see the entrepreneur's journey going from an idea to an iteration and then become part of the continuous improvement. So it's hats off to you and your passion. And it's always a pleasure to hear from you.
Thank you.
Right. Any questions from anyone? Feel free to chat.
Hi, my name is Dr. Benjamin Wang. I run a little medical device company here on the West Coast called NEVAP. You know, we're a little farther along in this process. We have an FDA CE mark cleared subglottic suction endotracheal tube, basically designed to reduce patients' time on the mechanical ventilator, but also designed to reduce post-operative pneumonia. Are we at a stage that's maybe a little too late for the IBE program, or is it, you know, because I'm always looking for additional opportunities to network and get our solution out there, especially during this time when COVID-19 is increasing the number of chronically ventilated patients.
Benjamin, thank you for your question. No, I don't think it would be, I think it would be very valuable to you for several reasons. I think the scope and spectrum of knowledge that we provide and insight and perspective we provide always provides an opportunity for people to learn, even those that have been in the industry for quite some time. Many of our faculty members are CEOs and, you know, have done 2 and 3 exits. And often they'll sit in on the presentations and say, geez, I didn't know that, or geez, I wish I'd had known that earlier, or I'd like to, you know, talk to that faculty member because I'd like to maybe bring them on as an advisor. So I think that the networking and the knowledge that you would gain from the process would be of value to you. I think it would also potentially be valuable to people on your team. I think Tony is a great example of that, how he has utilized the IBE as a catalyst to get people to think in a more entrepreneurial way and support their development and evolution. And we have a growing number of investors and other people now that are coming to audit and check out what we're doing. So I think on a couple levels, and I'm happy to talk to you about this in more detail, you know, after, you know, if you want to catch up with me afterwards. But yeah, I think it would be a very valuable thing for you.
Okay, thank you.
Mm-hmm.
Excellent. Let's see, I don't see anything in the chat. Feel free if anyone has any questions, speak up. Otherwise, I know it's right on the 8 o'clock, and we'll take a couple more questions, and then we'll be sending out a follow-up email with every speaker's contact information and the link to the recording of this webinar, so you can share with your peers and your network. But we'll take a couple more questions before we conclude. All right. So with that, I have a 10-second rule. I want to thank you all for joining and look forward to seeing you guys again. Thanks a lot again, Kurt and IBE team and all the speakers, Tammy, Tony, and Atmir for giving your valuable time. And we'll be following up with all the attendees. And thanks a lot for Insight and Ivan team to organize this webinar. As usual with COVID-19, this is our first 4 day of doing the complete virtual event. We have been putting our events live on Facebook. Join us at SoPE. And if anyone is interested in following up with any of the panel speakers, feel free to reach out to any one of us and we'll be more than happy to connect you. Take care.
Thanks.
Thanks,
a lot,
Drew.
guys.
Thanks, Arlen. Thanks, team. Take care.
Take care.
Thank you.